in Chapter 3 now, and in this chapter we're going to be focusing on the ability to study microorganisms. So to study microorganisms, we first have to know a little bit about microbes. All microbes have certain characteristics. All microorganisms, living organisms, have characteristics that are similar. All microorganisms and all organisms in general have DNA. DNA is the genetic material that carries the information of who you are. It is the code for all the proteins in the body and is composed of adenine, thymine, guanine, and cytosine. All living things have to be able to metabolize. Metabolism is the sum of the chemical reactions within your body cells. So bacteria, microorganisms typically are single-celled or very small, maybe double-celled, but if you're looking at eukaryotic organisms, but they're very small. And so within their individual cells, they're going to have the buildup and breakdown of different nutrients. All living things have to be able to reproduce, and so this is just a cute picture of a kitty cat and slow metabolism. I thought it was adorable, so I threw it on here. All living things have to be able to reproduce. So microorganisms, or I should say bacteria, reproduce via asexual reproduction called binary fission. If you're looking at eukaryotic organisms, they may reproduce sexually or asexually. Here is a cell, single cell. The DNA is doubling in that single cell. The DNA then moves to the opposite sides of the cell. We have a cleavage furrow forming, and then we have two identical daughter cells. That's known as mitosis. So eukaryotic cells can also replicate asexually. Living things also have to be able to adapt. An adaptation is just a change that allows an organism to be better suited to the environment. Living things have to be able to respond to stimulus and have to be able to be homeostatic or maintain homeostasis. So responding to a stimulus, I don't have a picture there, so apologies, is the ability to either move towards or away from some sensation. Think about touching a hot stove or drinking a hot cup of coffee. When you put your lips on the cup and it burns your lips, you move away from it. Or plants bend towards sunlight. Bacteria will move towards nutrients and away from anything that is going to be considered dangerous. And then homeostasis is the ability to maintain internal environment within certain variables. And so this is just a basic homeostasis, homeostatic mechanism. We have a receptor that takes in stimuli that tell us what's going on. That receptor sends signals to the control center. The control center determines what needs to be done. And the response goes out to effectors, and effectors are what bring about change and bring us back to homeostasis. So when you think about homeostasis, think about just maintaining health. Anything that brings you, and you're constantly changing. Think about right now when you're sitting. If you stand up, your blood pressure has to change to move or to increase with the new needs for the body for oxygen. When you're sitting, you don't need as much oxygen. When you stand up and start moving, you need more oxygen. That's homeostasis. When homeostasis is not being maintained, that's when disease occurs. Bacteria live in groups, typically known as multicellular associations. And these multicellular associations are often called biofilms. So when you think of a biofilm, don't think of a multicellular bacteria, because it's not. Bacteria are single-celled, they're always single-celled, and they can replicate, but each cell is an individual organism. What a biofilm is a group of cells that come together and they form an association. So they live together, which allows them to benefit from each other. And in the natural environment, about 99% of bacteria form organizations known as biofilms. And this is just like protection. Think about when you have a large herd of deer. Those deer in the middle are very protected. The deer on the outside are the ones that are potentially going to get hurt. And I guess deer don't really form herds, do they? Buffalo, they form herds. And so a biofilm is then a multicellular association which starts typically with an individual bacterial cell that adheres to a surface using its pili. Once it adheres, it starts replicating and it produces a chemical, a carbohydrate substance that surrounds the entire group of cells. Once that happens, then more cells can move into the environment. So we have bacterial cells adhering. This is a reversible attachment. Once that exopolymer gets on there, then or gets produced, then the bacteria no longer can be removed from the surface. Now we have new cells can move in. Bacteria can still replicate. They can gain nutrients from other cells, and as new cells move in, those new cells can produce nutrients that the cells on the bottom can use, and some of the waste products from the cells on the bottom might be nutrients that other cells can use. And so this biofilm is going to grow and grow and grow. And eventually, some of the cells may leave the biofilm and form a new biofilm of their own. When bacteria live in groups, they talk to each other too. So they don't have our language, but they do have the ability to send out chemicals that help them to determine the size of their community. They also can determine like if nutrients are available or not. And so if you look at the micro focus on page 74, it talks about how microbes can use either, and so the examples they're showing, myxobacteria, which form what they call wolf packs. These are a group of microbes that live together, and they send out chemical signals that help them determine if there's food or other bacteria or viruses or anything else around, and then they move towards that and they engulf it and eat it. The other example is Alivibrio fishery will send out chemicals that help them determine if there's enough cells in the environment, and if there are, then they will glow. They will basically turn on a protein. That protein stimulation causes the cells to bioluminesce. Two examples of quorum sensing. Here is a biofilm that most people are familiar with. This is plaque. The interesting thing about biofilms can form almost anywhere, but they cannot be removed except through mechanical removal. So you can use like Listerine to kill off some of those superficial cells, but the cells that are within that biofilm, deep in the biofilm, are not going to be killed off. The only way we get rid of the actual biofilm is by mechanical removal, so brushing your teeth, which is why you're told to brush your teeth twice every day. Because as soon as you brush, you're going to then have more bacteria starting to adhere to form a new biofilm. And this is just another picture of quorum sensing, which I talked about on the last page. Here you have individual cells, low density. They send out signals, don't have a lot of cells around. So once they replicate enough and have enough cells, then they can maybe alter their genes, change some behavior. Both prokaryotes and eukaryotes have similar organization. So one characteristic of all living things is we have to be organized. Prokaryotic organisms have a cell structure that is similar to a eukaryotic organism. In both cases, they have a plasma membrane. Plasma membrane surrounds the entire cell and allows that cell to maintain internal materials from the external environment. They have compartments within the cell. So over here we have compartments called organelles. So like you have the mitochondria, you have the lysosomes, you have a Golgi, you have a rough ER, you have smooth ER, stuff like that. Each of those structures has their own functions. Over here we have a region of the cytoplasm designated for the DNA. We also have little tiny structures called ribosomes, which are found over here as well. You have microcompartments that contain nutrients or gases as well. We're going to talk more about the structures found within the prokaryotic bacterial cell in Chapter 4. So instead, I'm going to get right into some characteristics that hopefully you remember from your eukaryotic organisms. So within a eukaryotic cell, we have organelles. Organelles have specific functions, so they're kind of like the tiny organs of the eukaryotic cell. So let's look at some of those organelles. The major organelle that everybody knows is the double membrane organelle called the nucleus, which contains a nucleolus right here. The nucleus contains the genetic material. The nucleolus is where ribosomes are formed. The nucleus has these tiny little pores. These pores allow the RNA to move out so that they can move to these ribosomes, which are found either floating freely or embedded in the rough endoplasmic reticulum. It's in the ribosome where we have protein synthesis occur. So in general, the RNA moves out and it moves into this series of channels known as the rough endoplasmic reticulum. This is where protein synthesis occurs on the ribosomes, and then we have modification of those proteins, maybe adding a carbohydrate group or a fat group to the protein. That protein is then going to be packaged up and shipped to the Golgi. So the Golgi is where we package up whatever the proteins are, and we determine where they have to go. So they're either going to go out into the extracellular environment, they're going to be embedded within the plasma membrane, or they're going to be released and become part of the cell's organelles. So think of lysosomes, peroxisomes. Here we have the smooth endoplasmic reticulum, which is continuous with the rough endoplasmic reticulum. This is where fats are produced. So we take in cholesterol and we can produce fats like testosterone or estrogen. A lot of our hormones are produced here. We also detoxify materials within these within the smooth endoplasmic reticulum. We have lysosomes and peroxisomes. So here you can see lysosomes. Where is my peroxisomes? Do I have a pretty picture of a peroxisome? There. So if you look, here's a peroxisome and here's a lysosome. They're very similar in structure. They have similar-ish functions. Lysosomes produce digestive enzymes, so they're like the digestive system, whereas peroxisomes produce oxidative enzymes that will kill anything. So lysosome will digest materials. If bacteria are brought in via endocytosis, then the lysosome will bind and digest that material. Peroxisomes will oxidize materials. They produce oxidizing agents that break down the cell structure completely. So if a bacteria binds with peroxisome, and the end result is hydrogen peroxide, which is then broken down further into water and oxygen. Peroxisomes also break down free radicals so that free radicals can't damage our cells. We have centrioles. Centrioles here are composed of microtubules. Centrioles are what produce the spindle fibers that allow DNA to move properly during mitosis and meiosis. We have the cytoskeleton. So centrioles are formed from microtubules. Cytoskeleton has 3 types of proteins. Cytoskeleton helps to maintain the shape of the cell. So it binds the plasma membrane. It helps to maintain the integrity. But it also moves materials around throughout the cell. I talked about it, so I think I'm good. Oh, the mitochondria. I did not talk about the mitochondria, did I? Mitochondria is the powerhouse of the cell. Mitochondria is where energy is produced. And then if you're looking at plant cells, so this is an animal cell, so you don't see this. You don't see plant cell materials. But in a plant cell, you would also see a central vacuole, and which carries water, holds a lot of extra water. You would also see a chloroplast, which contains chlorophyll, which traps light energy and helps us helps plants in producing food from sunlight, so photosynthesis. There is, and I'm going to go back really quick just so you can see it. Right here, there's a link to a short, maybe 5 or 6 minute video that goes over all the organelles a little bit slower than I just did. Eukaryotic and prokaryotic cells can both produce photosynthesize. Not all eukaryotic or prokaryotic cells can photosynthesize, but those that can do. They might use different protein trapping molecules. So cyanobacteria and plants and algae all use chlorophyll, but some of the other bacteria use different photopigment trapping materials. Both eukaryotes and prokaryotes have similar structures that form what we would call a cytoskeleton. You wouldn't call it a cytoskeleton in the prokaryotic organism, though. Here's a comparison of prokaryote and eukaryote. So you can see they both have ribosomes, they both have the cytoplasm, they both have a plasma membrane, they both have DNA, it's just different structure. Where these different functions occur. They both have flagella, we also have cilia. They can have a cell wall depending on the structure or the organism. Last thing I'm going to talk about is the endosymbiotic theory or endosymbiosis. What endosymbiosis is, two cells that come together and form a mutual, well, in this case, a mutualistic relationship. So the idea is that there are a lot of different types of cells. Some of these cells were large, and some of these cells were really small. The small cells and some of the cells were very specialized. So in this case, we have a small specialized cell that was really good at producing energy. Then we have this large cell. So the small cell was engulfed by the large cell, but instead of the large cell breaking down the small cell, it formed a mutualistic symbiosis with them. Over here we have a small cell that is really good at photosynthesizing. Same thing happens, it's engulfed, and instead of breaking it down, they form a mutualistic symbiosis. Symbiosis, they live together. Mutualism, they gained benefits from living together. So now we have this cell that has a chloroplast structure, so that can produce food, and then we have this structure here that takes that food and can break it down to produce energy. And this is kind of how we believe eukaryotic cells came about. All cells were prokaryotic, and eventually through mutualism relationships, we got these different organelles, and then eventually the DNA engulfed itself with some of the plasma membrane, forming that nuclear envelope, forming the nucleus. And now we have these different structures inside the cell, these different organelles. And what's really cool about these organelles is that if you look at both mitochondria and chloroplasts, they have their own DNA. The DNA that they have is circular, just like the bacterial DNA. They have two cell membranes. The outer cell membrane is different from the inner cell membrane. The inner cell membrane is just like the, what do we call them? The ancestral bacteria. They have their own ribosomes. So ribosomes are similar to bacterial ribosomes. So they have what they call 70S ribosomes versus 80S ribosomes. They can produce their own proteins because they have DNA and ribosomes. And if we take out the mitochondria or the chloroplast from the cell, that cell cannot produce its own mitochondria or chloroplasts at all. So those are a lot of proofs that this endosymbiotic theory is likely the correct or the mechanism at which we produced a eukaryotic cell from prokaryotic cells. This theory was proposed by Lynn Margulis in the 1980s, so it's a very relatively new theory, and it's the best theory we have right now on how eukaryotic cells came about. I am going to stop here because the next video will start up on this slide. So you guys have a great day. Bye.